Good morning, good afternoon. I want to welcome everyone to the Protocol Deviations Management Toolkit session, our webinar from Transcelerator. My name is Leslie Ware. I am a program manager for Transcelerator, and I'm happy that you all are able to join us. On the call today, we have Katherine Stuart. She is the co-lead of the Protocol Deviations Initiative here at Transcelerator. She's with Pfizer and located in the UK. And also Laura Gallucci, who is the Director of Global Clinical Trial Operations. She's with Merck and also located in the US. Additionally, we have Amy Janowski, who is our Transcelerator Project Manager, also located in the US. So before we get started, I would like to just go through a few important aspects of participating in the webinar. First off, all participants will be muted for the call. One nice feature is that you can minimize the control panel that you see on your left-hand side by clicking on the orange button, and it will allow you to see the full screen. To ask a question during the presentation, there is a questions tab you will see on the control panel. We will be answering questions during the session and also at the end, so feel free to type your question. Don't forget to press send. Just for awareness, this webinar is being recorded, and we do plan to make the recording available to everyone following the call. If you have any other questions or technical issues, you can email events@transcelerator.com. We will have live polling during this webinar. All polling is voluntary and it's anonymous, but what I'd like to do at this point is be able to test it. So I'm going to test it by asking you what your favorite color is. And you can go to that poll question that will show up and you can click on that color. So I'm going to launch that now. And if you would click and just test that it's working. Great. I'm seeing about 60% of you have voted. So I'm going to go ahead and close the poll. Great. Thank you, everyone. It looks like everybody's favorite color. It looks like it's blue with a nice variation of the other three colors. Okay, just a few ground rules. We want to make sure that this discussion is helpful for everyone, and we're going to try to answer as many questions as we can. So let's do a couple quick ground rules. First off, participation in the questions is voluntary, as is using any of the Transcelerator assets and tools. When you are asking a question, you don't have to identify what company you work for. There are a few things that we do ask that you don't ask as a question or discuss, and that's around vendors, sites, and CROs that you're using. Any issues you might have with those vendor sites or CROs. Please don't share your long-term development plans or anything related to cost. And just for awareness, there are certain questions we won't be able to answer, and those center around any questions around vendors or the cost of using or implementing any of the Transcelerator assets and tools, and also which of the member companies might be using any of our assets or tools. So this is the agenda for the webinar today. I will give you a quick overview of Transcelerator. We'll follow that with Kath giving you an overview of the Protocol Deviations Toolkit that's been developed. We are seeking your feedback and public feedback on that toolkit, and then we'll follow it up with Q&A. So before we get started, I'd like to ask you your first polling question. How well do you understand the ICH definition of an important protocol deviation? Little to no understanding? Do you have an intermediate level of understanding, or do you have an advanced understanding? Okay, it looks like, I'll share this in a minute. It looks like we're looking at about an intermediate level of understanding, followed by a little bit of an advanced understanding. So I think most of us have voted. I'm going to close the poll, and I'm going to share the response with you. So it looks like we're about at an intermediate level of understanding as the majority of those attending. So let's move on. Okay, I'm going to give you a quick overview of Transcelerator for those of you that have not really understood much or have engaged with Transcelerator. Transcelerator is a not-for-profit organization. We're in the pre-competitive space, and we were created to foster collaboration, really focusing on improving the health of people around the world and accelerating and simplifying R&D. Transcelerator was started in 2002. We had 10 member companies at that time, and we started with 5 initiatives. Since then, we have grown to 20 member companies. We have over 25 initiatives. We operate in 22 plus countries, and we have engaged with around 13 to 14 health authorities around the world. This is gives you a visual on who the members are at this point. It is available to the biopharmaceutical research and development organizations. And our membership really is in these individuals from these companies. Transcelerator itself is a very small organization that is designed really to facilitate, and our initiatives are built and developed.
Leslie? Leslie, I think we lost you on audio.
Okay, well, so go ahead.
Thanks. This is Katherine Stuart speaking. We seem to have lost Leslie's audio, so let's move on to the presentation itself about the Protocol Deviations Management Toolkit. So this is Katherine Stuart speaking. I work for Pfizer in the UK. I'm one of the co-leads of this initiative, and I'm also joined by Laura Gallucci from Merck in the US, who is the other co-lead. So I'd like to start out by saying a really big thank you to all of the member company representatives that worked with us on the Protocol Deviations Initiative. We had a cross-functional group of people from, I think, 11 or 12 of the member companies. So we had good representation from clinical, from operations, stats, data management, quality, and that enabled us to get a really wide range of perspectives and inputs as we moved through our process of thinking about protocol deviations and what truly matters. So I'm going to spend the next 30 minutes or so taking you through the work that we've done and the materials that we've developed. And at the end, I'll be showing you how you can go onto the Transcelerator website and access those materials. And we're seeking as much feedback and input as we can. So it will be great after this presentation if you can go off, have a look at our toolkit at your leisure and let us have your comments back. Also, thank you to those of you that have submitted questions to us in advance. I'm going to try to cover as many as possible of those as I go along. And Laura is going to be manning the question and answer panel as well. So we hope to be able to make this as interactive as we can, with the caveat that you are all hard muted, I'm afraid, and can't ask questions verbally. Okay, so with that, can I have the next slide, please? Okay, thank you. So the Transcelerator Protocol Deviation Initiative started way back at the start of 2018 when we started talking to each other, and we realized that dealing with protocol deviations really poses a challenge for many member companies, and the guidelines that are available to us to follow are fairly top level. They're not particularly specific. You know, we all want to do the right thing, but it's really hard to know that you're doing the right thing if you haven't got much guidelines to follow. And we came to define our problem statement as the fact there is this lack of clarity as to what exactly is an important protocol deviation. And that's led to both overinterpretation and underinterpretation, which we think could have quite a material impact on the ultimate management and reporting out of important protocol deviations. So we felt this is really an issue because if we have a lot of noise in the process, you know, it gets in the way. If you've got a huge volume of substrate to go through, perhaps you are going to miss something that truly matters, or perhaps it will take you much longer to find what truly matters. So we started to think we'd like to be able to come to a position where we can confidently look for important PDs, define them in a cohesive and logical and pragmatic way, and really focus on what matters. So the impact statement we have is that over or under interpretation of what's an important PD can increase the noise in the system. So like I said, slowing us down to look for what truly matters in terms of safety or data integrity, and potentially influencing our trial results and the protection of our human subjects. So the other thing we were wanting to do was to try to make sure we're incorporating the ethos of ICH E6R2, which is really mandating all of us to do everything using a more risk-based methodology, thinking carefully before we start about what matters, and then focusing our time and attention on that. So, that's what we're trying to do with PDs, making sure we spend the effort on the important stuff and be really totally sure that we're going to identify it in a timely way. So next slide, please. Okay, so we started off with a survey of the Transcelerator member companies, and you saw who they were on Leslie's slide a few moments ago. And one of the first things that we found was there's actually this huge variation in terms of the vocabulary that we're using. You can see this word cloud here. I alluded before to the fact that there's not that much in the guidelines about protocol deviations and what they really are. And here we were, the Transcelerator member companies, using important or not important, using major and minor, using deviation or violation. Some companies had a critical classification. So it was actually very confusing. And we were all using different terminology to talk about the same things. And that confused us. And we then postulated from that, well, if this is what we're doing internally within our companies, perhaps that confusion spills out and causes challenges out in the field. So if I could have the next slide, please. Thank you. So we expanded our survey and we talked to investigational sites and we talked to some IRBs as well. And the responses that we got back, confirmed this suspicion that we're all using different words, using different definitions, and it's really confusing because our investigational sites obviously work with more than one sponsor. IRBs obviously get data submitted to them from more than one sponsor, and we're confusing them with the terminology that we're using. And that really could matter. Perhaps we're distracting people from concentrating on work that's important. Perhaps we're not getting things reported in the timely way that should be. So really, there must be a sweet spot where we can start to align around the terminology. So in conclusion, the whole PD field seems to be pretty complicated. It's varied in terms of how companies are handling it. And we're using different definitions in terms of important and critical and major. What do they even mean? And that leads to process inefficiencies. So sites struggle to comply with different requirements from different sponsors. Within each sponsor company, there tends to be a lot of churn and a lot of man hours spent on managing PDs. And we felt that perhaps there were, some proposals that we could make that would make this whole process more efficient and more truly focused on what matters. So next slide, please. Okay, so like I said at the beginning, we got together at the start of 2018 as a Transcelerator work group, and we came together and we developed these objectives that we wanted to work towards. So we wanted to be sure that we could reduce noise in the system, meaning that we can quickly and confidently identify important protocol deviations. Again, focusing on what really, really matters and not being distracted by things that come into our system that probably aren't protocol deviations. Now, I'll talk about that in a moment. And then secondly, as well as having this laser-like focus on the important things, the impactful PDs, how can we actually be more efficient in handling these things, both from a site perspective, because they should be there taking care of the patients and making sure their data collection is as accurate as it can be, not struggling with our forms and our requirements. And how can we as sponsors also be more efficient, because that brings benefits to the study as well. If we can be truly sure that every man hour we spend on PDs is a man hour that is adding value. So we were planning to achieve these objectives through three main approaches. So firstly, to going back and looking at what the guidelines say and proposing an improved interpretation, including the definition of important protocol deviation. We want to propose improvements in the management of protocol deviations, like I said, to make it more efficient, to focus on what matters and be sure that we are being accurate and consistent within an individual company. And then finally, and most importantly, we wanted to take every opportunity we had to engage with FDA and with other health authorities on what we see the problem statement as being and how we think some of the challenges we've recognized could potentially be addressed. As I said before, looking more at things with the flavor of ICH E6 R2, so truly being risk-based and focusing on what matters. Okay, so if we can go to the next slide, please. Thank you. All right, so what we went back and did was we looked at, obviously, the definitions we have in ICH of what is a protocol deviation. So ICH E3 is the CSR one. That talks about protocol deviations in terms of important PDs in the CSR. And then in the ICH E3 Q&A, it also gives some further examples and definitions. And in terms of the definition of protocol deviation, it says, and I'm quoting from memory, a protocol deviation is any change or divergence from what the protocol says. And actually, we discussed this quite a lot in our group, and it became apparent that actually using that as is not always meeting all of our needs. And we all had lots of examples and discussion points about how we struggle with that. So we've proposed 3 more factors that we think would also need to be present to mean that something is a PD. So the first one is that the event actually occurred. So it's not a theoretical risk of something that may happen in the future or something that could have happened if something else had happened first. It did actually happen. So a protocol deviation can only be a true occurrence. The second one was that the protocol deviation must be something that's related to either a data point in the protocol or a process that's specified in the protocol or in one of the protocol reference documents. So again, we had a lot of discussion, and I've got another slide later, about monitoring issues. Many observations that CRAs make at sites are completely genuine, completely accurate. Sometimes they come through as a protocol deviation, and actually they are not a protocol deviation because they're not referring to something that is specifically mentioned in the protocol. And then the third additional definition that we decided we had to add was that protocol deviations should be reported independently of fault, blame, or circumstance. So the point of this is it doesn't matter whose fault it is or why something was not done in accordance with the protocol, it needs to be collected and analysed anyway. So we had discussions about, well, the site didn't mean to do it, it was the patient that did it, or the visit was performed out of window because the site was closed due to bad weather. That's no one's fault, so that's not a PD. But we decided that we're not collecting PDs specifically to look at blame or performance. We're collecting them to be sure we've got a really accurate and complete picture of any instances where we didn't follow the protocol as written. So reporting PDs, regardless of why they occurred, is a really very important factor that we want to highlight here. So the other thing that we wanted to do was to look at actually what important protocol deviation should mean. And here is where we really brought in the flavour of ICH E6 R2. So we're proposing a definition of important protocol deviation is obviously a subset of the overall data set of PDs, which may significantly impact key study data. So that the word key there is the important word, the word that we've added. So potentially if you have a study with an exploratory endpoint or an optional sample and something is not done correctly with either of those things, that would probably be a protocol deviation, but would not be important because it's not impacting your key study data. So again, trying to really focus down on what are the important things for this protocol, what could impact the safety of our subjects, what could weaken the integrity of our data. Okay, so next slide, please. Thank you. So here are some examples of protocol deviation. These are all true examples that have been reported into different member companies through their respective protocol deviation management cycle, whatever that might be. And, you know, these kind of look funny. I think some of them are quite funny, and we had a laugh over some of these. But the point of this slide is that if you're dealing with, 100 of these a day, it's going to slow you down in terms of identifying the patient that went in without being consented, or the patient that has become pregnant during the study, or whatever it might be. So all of these things, I think, potentially could be something the monitor would want to document and follow through the monitoring visit log or issues management system, but not really protocol deviations. I mean, the informed consent was stapled multiple times. Who cares? It's not a PD. It's not impacting on subject rights. It's not impacting on something that's stated in the protocol. So it's just a fact, an observation. And we want to try to improve people's focus so that things like this do not get reported as a PD. So you can see on the right-hand side, we have a decision tree, and this actually appears in some of the materials that we've developed. So you start at the top in the top gold and diamond. So is it a protocol deviation? Did this thing actually happen? And is it related to a data point or process in the protocol? And if no, you stop, and perhaps you manage that particular item through an issues management system, but you do not take it any further through protocol deviations. But if yes, if it is related to a data point in the protocol and it has occurred, yes, it's a PD. So then you move on to thinking about, well, is that an important protocol deviation? So does it impact key study data or the subject's rights or safety? Yes, it's an important protocol deviation. No, then by definition, it's a non-important protocol deviation. And it's important to say here that things that start out in their life cycle as a non-important PD could later on become important. So if you have a whole host of non-important PDs relating to the same thing, then you've got a trend. That is probably something that you're going to want to address. If you get enough of those things, perhaps you meet your predefined threshold, and then those do become important protocol deviations as you go on. So classification of important versus non-important can change as a trial progresses, and it's important as a study management team that you're keeping very close tabs on your trends and how things are playing out, because you obviously want to react as quickly as you can when you start seeing a signal. Okay, next slide, please. All right, so those of you that have seen some of the other Transcelerator materials before from other work streams may well recognize this issues management function. So this is all from the quality management system framework, and this is just a way of managing issues. So when you come to look at your PD and you're making a decision about whether it's important or not, it's not always going to be the same. That's why this is tricky, because it may depend on what is the investigational product that you're working with, what's the patient population that you're studying, what's the endpoint in your trial, how long is your study, is it pediatric? There are many things that you need to take into account. But our whole approach is built around thinking about those things carefully before you start, having a good prospective analysis of where your risks are, and then obviously writing your protocol to minimize those risks and predefining where you think your important protocol deviations are going to be, also in relation to those risks. So you can see in the picture on the right-hand side, there's a whole host of bubbles going in at the top, issues, observations, but they go into the funnel, and then you apply our protocol deviation pathway that I showed you on the previous slide. Some of the extraneous things get filtered out, and then in the bucket at the bottom, you're left with the issues that matter. So your protocol deviations and your important protocol deviations. You've been confident and methodical about how you've got to that subset. You've been confident and methodical about the definitions that you've used, and then you've got there quickly, and you can be really focusing on risks to your data, risks to your subject in a timely way. So how do we propose that we do this? Next slide, please. Okay, so what have we actually been doing during our work in 2018? So I'll show you how we've engaged with FDA in a later slide, but in brief, the team have developed 3 separate components of the Protocol Deviation Toolkit. So the first item that we have is the PD process guide. So this is a Word document. It's about 30 pages in length. It describes our proposed interpretation of definitions, and then it describes the life cycle of a protocol deviation. So in terms of how protocol deviations are identified, and there's, I'll talk later, different ways of doing that, how they should be classified, how they should be categorized, trend analysis, how you review these things on a regular basis, what you do with signals that you find, how do you report them out in your CSR, and then following that, what closeout activities are required. And it also covers things like iterative training, iterative other actions that you may need to take. So perhaps you need to go back and look at your protocol wording if you're seeing lots of PDs around the same item. Perhaps there's site training issues or CRA training issues that you need to look at. So it's a very cyclical process, and you need to be sure that you're taking all your learnings and emerging signals and feeding them right back into the beginning again. So the guide talks you through that whole life cycle from start to finish. Then the second component is the map. I'll show this to you later. This is just a 1-page visual of the life cycle of the PD. So rather than having to read 30 pages of text, it's showing you how each step of the process fits together and where the feedback loops come in. So if you're a visual learning person, that's a really nice component to have. And then the third piece of the puzzle is the PD assessment tool. And this is an Excel template. And this is designed to enable you to apply the processes and definitions that we've provided in the guide and the map, and ensure that you consider those in a methodical and consistent way. And many companies have their own systems and tools to doing that, and that's absolutely fine. This is just an example of how it could be done in an Excel format. So these 3 things taken all together comprise our PD toolkit, and it's these 3 items that we're seeking public feedback on at the moment. So I hope very much that you'll all go away after this webinar and take a look at those. Okay, next slide, please. Okay, so as promised, this slide is talking about the engagement that we had, particularly with FDA through 2018. So as I mentioned before, we came together as a work group really at the start of 2018, and we continued working together pretty intensively until the middle of the year, and then slightly more sporadically in the second half of 2018. But we were very fortunate in that we had a number of opportunities to engage with FDA, to talk to them about the challenges that we feel that we face in this area and what some of the proposed solutions could be. So if we look at the arrows at the bottom here, this all started with a lunch and learn with FDA back in February. We were then able to have an orientation meeting with a number of FDA representatives in May. We had some further follow-up in July, and then in October, FDA sent us a list of questions and comments on the materials and the interactions that we'd had at that point in time. We regrouped as a work group to consider FDA opinions. We made some updates and provided some clarifications, and we sent all of that back to FDA in December in 2018. So next slide, please, Leslie. Okay, so now we're into 2019. So in Q1 of this year, we had feedback from FDA that they had no further comments or questions on the materials or responses that we had provided to them, which was great news. Following that, we had a further review period with the Transcelerator member companies. So I mentioned at the beginning, we had representation on the work group from most member companies, but not all. So we had an internal review period where particularly those member companies who hadn't had representation on the original group were able to look at the materials, ask questions, provide feedback. Any updates to things were updated following that feedback. And now here we are in Q3, Q4, and we're looking for public feedback. So from really any interested party, be it non-Transcelerator member company, site, IRB, whoever you may be, we're very happy to receive feedback from you. And that will close in November, in the middle of November. Following that public consultation, we'll take all the comments and questions on board. We'll look at whether any updates or clarifications are needed. And obviously, we're very open and willing to engage with other health authorities as well. We're actively pursuing a number of opportunities there. All being well, then the intention would be that we would finalise the materials and make them available on the Transcelerator website from Q1 next year. So that's the plan. Okay, so enough of the preamble. Let's have a look at some of these things. So next slide, please, Leslie. Right, so this may look small. This is the map. So what you're seeing on the screen now is the life of a PD. So when you come to do your study, as I said before, planning before you start is absolutely key. So make sure before you start that you're thinking critically about where are the risks. So in terms of what's in the protocol, what risks does that bring? In terms of what's in your IB, what risks does that bring? In terms of your RACT, so your risk assessment classification tool, what have you identified already that is a risk? So make sure you're gathering input from all of those sources to carefully plan what could go wrong, how can you stop it? If it does go wrong, what are we going to do? Make sure all that thinking is done before you start. Then you get ready for your particular study. You develop a prospective list of what you think your important protocol deviations could be. You start populating your protocol deviation assessment tool, and then off you go. So you'll see in the first yellow vertical bar, you've got a little red explosion going on. That's your PD happening. So you can identify that through programmatic or manual means. It's reported centrally. You decide whether it's important or not important. You think about what category it goes in. So what label is it in? Is it to do with inclusion/exclusion criteria? Is it to do with investigational product? So you can start to put these things into smaller buckets, and that enables you to do a more powerful trend analysis. Then you can see the pathway splits. So important PDs are managed along the top, and as you would expect, more happens to them. They're important. We should spend more time on those. Those are really impactful. We need to focus our efforts on the important ones, learn from them, minimize them, and take any other actions we need to safeguard our subjects or the integrity of our data. Important PDs go along the bottom pathway. That's less labor intensive because we've been consistent and confident about our analysis of what's important and what isn't, so we can spend less time on those. So they both travel along from left to right. We do some periodic reviews, and then ultimately we report important PDs in the CSR, we archive everything, and we move on. So that's basically the life cycle of a PD. It takes us 30 pages of text to explain that, but there it is in a one-page picture. So now I'll take a couple of slides to talk in a little bit more detail about these major steps, and then I'll show you how you can access materials. Thank you. Okay, so the life cycle of a PD is broken down into really 7 stages. So in preparing, like I said, you're thinking before you start, what could go wrong? Looking at your protocol, your IB, your SAP, your RACT, really understanding what the risks are. And we've also incorporated some of the guidance from the common protocol template. So don't write a really complicated protocol with very stringent conditions if you don't have to. Don't say the visit window can only be 2 days if actually 3 wouldn't matter. So obviously the more complex your protocol is, the more PDs you're going to have. And we recommend that, again, you use all of this risk analysis to inform the writing of your protocol. The other thing that we've proposed in the prepare section is that you think about important PDs in terms of a hierarchy. So There may be some that your organisation will choose to say, if this happens in any trial, this will always be important. So for example, it could be if a subject enters the study but has not given any consent. Perhaps you would have that as your organisational level important PD. And then every study that runs in your company will have that same definition. And then you're sort of having intra-company consistency, and you're not having to reinvent the wheel every time. So we've proposed that you consider an organisational level definition of IPD. Then we take a step down and say, well, perhaps there would also be an intermediate level important protocol deviation. And this may be in terms of a particular medicinal product. Perhaps it's infused. So there would be things around the infusion rate or dilution that would always be important if they weren't followed. So perhaps you want to put that in place so that within your suite of studies with that particular asset, you have consistency. Again, each individual team doesn't have to make their own decisions. All that work has been done beforehand and they just inherit that and run with it. Perhaps you might think about that in terms of patient population. So for example, with a pediatric study, could there be particular things with the pediatric population that you would always want to define as important within your company? So that would be like the intermediate level, and then we take a further step down to the study level. So when your study team is starting, they will inherit some of these organizational level and intermediate level definitions already. So they don't have to rework those. That's been done for them. What they need to look at is this specific protocol and what are the nuances here that we haven't captured already. So perhaps there's a different comparator drug. Perhaps it's a much longer study than all the others. Perhaps it's got a different study procedure in. So those bring new risks, new opportunities for things to go wrong. And if your study team can focus on what's different about this protocol and be confident that they're being consistent with the protocols that have gone before, that's going to save time and also ensure that you still have that laser-like focus on what genuinely matters. Somebody else asked a question actually about quality tolerance limits and, could number of PDs potentially be a QTL? How do we set these things? And there is actually a separate Transcelerator work stream on QTLs, which will be putting out materials, I think, in the fairly near future. So QTLs, I think, is something everyone's still bedding down within their companies, and there should be more coming out on that in the near future. So moving on from prepare to train. Training is obviously really important. So there's different training appropriate for different parties. So the study team obviously need to know the protocol, and they need to know the contents of your protocol deviation assessment tool. So what have you decided is important for this study? Where are the risks? What are they focusing on? The sites don't need to know the content of your protocol deviation assessment tool. They need training clearly on the protocol, the procedures, the vendors that you're working with. They need to be confident that they understand the protocol and can execute it as it's been written down. As the study goes on, you may find that emerging signals mean that you need to loop back and retrain one or both of these parties, depending on what you see. Okay, so we've planned and prepared, we've trained. Now we actually start. So we need to identify PDs and we need to collect them. In terms of identification, they can be identified through programmatic means, so straight from the study database, or they can be identified through manual processes. So the CRA at site looking at things, or potentially a central monitoring group looking at things. So there's different ways these things can come in. We've recommended that programmatic identification is used wherever possible. This is obviously more consistent. It's not down to 20 different CRAs trying to do the same thing, but perhaps doing it in 20 different ways. Particularly for important PDs and those things that are numeric, there's no reason why these shouldn't be done programmatically. And the other piece to add to that is obviously when we start looking at risk-based monitoring, the monitors aren't looking at absolutely everything. They're not doing 100% SDV anymore. So it may well be that some non-important PDs do not get identified through manual identification. We accept that. But if the risk-based monitoring has been set up in the right way, we know that we are focused on what matters, the important things, and we should be picking up everything that's important. So once they've been identified, then the PD is classified. So is it important or not? And it's categorized. Which little bucket does it go in to enable us to do trend analysis as we move forward? Okay, next slide. All right, so the next thing that happens is the confirmation step. So we've found our protocol deviation, we've collected it, then the next step is to confirm it. So the confirmation step we recommend is done by a member of the study team who is not the identifier. And it seems that in many companies where CRAs are finding protocol deviations, they find them, they send them in, and then it's a member of the central study team that's doing the confirmation. And that's also quite important because if you have CRAs entering things, they only have the view of their set of sites, whereas central study team should have that more holistic view over the study as a whole, and is probably better placed to recognize, hey, there was another site in a different country that had the same sort of problem, and make that sort of link and spot the emerging trends a lot quicker. So the other thing that's covered in the confirm step is reconciliation of protocol deviations. So I know there were some questions about this. So the recommendation that we've come to with reconciliation is that important protocol deviations are reconciled. And by reconciliation, we mean going back to whatever PD management system you have and removing any duplicates. So if you've had a PD that's been identified manually and reported, and then the same PD has been identified programmatically and reported, you don't want that same important PD to be there twice. So we recommend that you check that and take out one of the duplicates, so it's only reported once. And we also recommend that the contents of that important PD, so what it says, is aligned to what's in the clinical database. So again, that we can be confident that all of the data we put forward on important PDs is accurate, is counted correctly, and is in agreement with what we have in the clinical database. All right, so following that confirmation step, we come on to reviewing and analysing. I've talked a bit as I've gone on about trend review. It's really, really important that this is done in a timely way. So PDs are reported in to the central team as rapidly as possible, that people are looking at the accumulation of data regularly. We recommend you set an agreed cadence for your review cycle, so it is done regularly, and that any emerging trend is promptly escalated to wherever it needs to go and actions needed are put in place. So retraining, protocol wording, whatever it might be, that can be put in place in a timely way. Okay, finally, reporting. So there's a variety of periodic reporting that may need to be done on a local level. So for example, some local IRBs have particular requirements about reporting. There may be local health authority requirements that you need to provide reporting for. That's fine. In the CSR per ICH E3, important protocol deviations are included. Okay, and then finally in the closeout section, so once the CSR has been completed, then obviously you archive your study materials. Again, different companies have different tools and processes for handling PDs, perfectly fine. We recommend that, obviously, like the clinical database, that PD data can be reproduced and provided for inspections if it's needed. Ditto monitoring issues and actions if some things have been filtered down to that pathway, and that a thorough lessons learned session is conducted so that other teams can benefit from learnings from that particular study. And it may be that the learnings from that go back to the RACT, it may be they go back to intermediate level or even organizational level important PD definitions. So making sure that you're constantly improving and refining what you have. Okay, so my penultimate slide, I think, or ultimate slide actually, what you've all been waiting for. If I can have the next slide, please. Okay, so here it is. So here on the screen is the route by which you can access the toolkit, the guide, the map, and the assessment tool. The comments on this will close on the 11th of November. So you don't have very long to download these and provide your feedback, but we really would welcome as much feedback as you can provide. We want to make these materials as valuable and as relevant as they possibly can be, so that we can be sure that we've provided a potential methodology for managing PDs in an efficient and accurate and a quality way. So with that, I think it's back to Leslie to tee up another poll.
Right, so we have one more poll. Let me launch that poll for you. Okay, how well do you understand the ICH definition of important protocol deviations now? And I just also, while we're collecting these responses, I just want to remind everyone, if you are putting a question in, please do remember to hit send. Otherwise, we can't see your question. Remember to hit send. Okay, great. I'm going to close the poll. We have about 72% that have responded, and I'm going to share the response. So it looks like we've had a definite shift from intermediate to an understanding. So thank you for responding to that poll. All right, we're going to open it up for a Q&A to Kath and Laura, and I know they've been monitoring your questions. So Laura, and I'm going to turn it over to you and Kath to perhaps pick out a few questions that you've seen that you want to share with this group.
Hi, this is Laura. I've been trying to answer the questions in the chat as we go along, but one that is worth speaking to, I think, is about pre-identifying what components would warrant being a important protocol deviation versus a non-important protocol deviation. And then, Kath, can you talk a little bit about the circumstances in which a non-important protocol deviation may actually become an important protocol deviation?
Oh, sure. So I think one of the things that we spent quite a lot of time talking about was, exactly that. How do you set your threshold and how do you know? So it may be, for example, that if one subject misses their blood sample at visit 3, well, perhaps that's not particularly impactful to your study data. But if every subject is missing that blood sample, then that's the whole, you don't have that endpoint to report for the whole study data set. So perhaps that's something where you would see this trend emerging and you'd think, well, actually, what's wrong here is a protocol misleading, have the central lab sent out the wrong kit, and you'd want to take some action there to evaluate that and put it right. And it may be that would become important if that's genuinely impacting the way that you can report that particular data point for the study as a whole. So that's one of the examples that we came to. Have you got anything to add to that, Laura? Other examples that we had?
Yes, in the question, it was the person asking the question was asking about windows, and they also asked why we would be recording something that was a protocol deviation if it did not impact the analysis. And if we go back to the definition, one of the items is about understanding the non-important, just like Kath mentioned. Additionally, going back to the definition that we talked about in the beginning, when it's a protocol deviation is independent of fault, it could be something that the sponsor may need to be aware of, so that potentially the sponsor process could change, or potentially there is a protocol amendment because the sponsor cannot actually adhere to a component that they were anticipating within the conduct of their trial as well. So all protocol deviations may not directly just impact the sites, but the sponsor may want to consider their own internal processes as well. Okay, I'm just glancing through some of the
Yeah.
questions here again. There's quite a few questions about the reconciliation process and confirming it. Can you talk a little bit more about some of the questions in that area?
Yeah, so I think it's fair to say that companies have very different approaches to programmatic PDs and reconciliation, and we did spend a lot of time talking about this. So again, the approach that companies take to programmatic identification varies widely. Some companies do it for pretty much everything. Some companies don't seem to do it at all. We came to the point, I think, where particularly with risk-based monitoring, we are recommending more programmatic identification because it comes off of the clinical database and is less open to individual monitor interpretation. And we also talked a lot about, well, how do you reconcile that? But we felt it's very important that important protocol deviations were fully reconciled so that when we put forward our listing of important PDs, that they will stand up alongside the clinical database. There won't be anything there that isn't mirrored in the clinical data. There won't be any discrepancies in terms of, well, the clinical database says this, but the protocol deviation text says that. So how do you as a CSR reviewer make sense of that? So we did recommend that important PDs do get reconciled against the clinical data to be sure that we know which is correct. If there's a discrepancy, well, actually, is it that we need to send a data query to the site to amend the clinical database, or do we need to have the CRA make an update to what the protocol deviation system is telling us? I'm trying to read the question as I go, Laura. Did that answer that one, or do I need to go into more detail?
I think you've answered that one.
I think the other, with risk-based monitoring as well, we're not going to capture every single PD because the monitors aren't looking at everything. So if you have programmatic checks in place, you can be confident there that you'll pick up whatever can be picked up programmatically. So things that are mathematical and fully objective and logical can be picked up in your programming. And you may miss some things that you can only find through going to the site. But again, if you've teed up your RBM correctly, you should be picking up what matters, and the things that you miss should, by definition, be non-important.
So, Kath, can you talk a little bit more about reporting of protocol deviations to ethics committees or regulatory bodies?
Yeah, and it's very difficult, isn't it, to be prescriptive about this because they appear to have, in terms of some interim reporting that different committees or agencies have, they can ask for what they like. Obviously, our key recommendation is that protocol deviation management is a living thing. You don't leave it till the end of the study and then do it in a panic, just before database lock. You should be keeping on top of the emerging data and issues as you go, so that if you do get an ad hoc request to submit something during the life of the study, you should be reasonably ready to pull that out of whatever system you're using and send that in. Some of them, we believe, do want to have non-important PDs as well. As we know in the CSR, we only need to include the important ones. And then there's additional local regulations. So, for example, in the UK and Spain and other countries, they've got the serious breach regulation where you have extremely expedited timelines for getting things submitted. But we didn't really delve into the nuances of local or national requirements. Obviously, if there's a requirement like that, you as a sponsor would need to comply with it in full in the way that was requested, and that's really outside of the scope of the toolkit that we have developed here.
Thank you. Can you talk a little bit about how there can be a harmonized approach across therapeutic areas by using the toolkit?
Yeah, that's a really interesting question. And we enjoyed talking about that a lot, I think, in the group. And I think what seemed to be quite a commonality was that every time you start a study, your team start with good intentions. They want to do the right thing. They want to be professional and consistent. And they can do either one of two things. They either start with a blank sheet of paper and build it from the ground up, which is great, but that takes time. And at the start of a study, you don't have time. there's no time. So that's not optimal. Or, perhaps they don't do things in a thoughtful way. They just kind of throw something together, and that's not optimal either. So we're trying to think, well, how do we save teams time when they're really busy, but how do we give them the building blocks to be sure that what they're going to run with eventually is a quality product? So then we started to think about, well, there are commonalities. And in fact, there was anecdotal input from somebody that they'd had an inspection finding where study team A had said that this and that and the other was important, and study team B, working on the same product, had said, well, this and that was important, but the other wasn't. And so there was a discrepancy between how they had defined protocol deviations, and they weren't able to adequately explain why they'd arrived at different decisions. So thinking about that, how can we enable teams to be consistent and to use the best knowledge? And that was where we came up with this hierarchy. So probably at an organizational level, there are things that will be important to every study. I mentioned consent. I mean, consent is common to every study. So perhaps there will be some issues of informed consent that you would define in your company as always being important. And then stepping down a level to either potentially a population level, so perhaps pediatric studies or perhaps PK studies. Perhaps there's commonalities in PK studies where you could always say, if such and such a thing happens, it's always important. Or within an individual asset, perhaps you're treating an oncology population, perhaps your endpoint is a 6-minute walk test. Perhaps you'll be able to say, well, for our company, if such and such happens in a 6-minute walk test, that would always be important. And then every study team that's using that endpoint or that indication can draw from your question bank, make sure that they're applying things in a consistent way. And then that's thinking they don't have to do. They can just take that, populate it into their PDAT and add in study-specific differences. And then you've got best practice, you've saved time, you've given intra-company consistency. And we think this will be a very exciting way to work. And of course, the other thing is that as learnings come in from individual study teams, they can be fed right back in at the top. So you're always taking advantage of the current state of knowledge within your own company, and you're ensuring that best practice is kind of forced down into all the study teams that follow on. So that, to me, I think that's a really exciting approach. I really like that.
Great, thank you.
Kath, thank you so much. We are almost at the top of the hour, and we have 2 poll questions for you before we end.
I'm sorry.
No, that's good. I'm going to launch the first poll, which is, as a result of participating in this webinar, would you recommend this information to a colleague? So we're about at 30%. Please take a few minutes or a few seconds to respond to this poll. And we have a second question for you as well. Great. So we're about at 60–70%. So I'm going to close the poll and share the results. So resoundingly, you have indicated yes, you will. So thank you for that. We have one more question for you as well. How likely are you to actually recommend this or explore the full toolkit, Protocol Deviations Kit? Remembering that the toolkit is out there on the Transcelerator website for you to comment on, and our date is the 11th to get feedback, 11th of November. All right, so we've got about 60%. For the most part, everyone is very likely to respond or to explore the toolkit. So that's great news. I'm going to just thank our participants and encourage you again to visit and comment on the toolkit. It is out there, and we really appreciate any feedback that we get. It helps us make that stronger. So as a reminder, we will be providing a link to a recording of this webinar later this week. So this concludes this webinar, and thank you everyone for your participation, and thank you to Laura and Kath.
Thank you.
Thank you.
Thanks, everyone.